Good evening. Good evening, colleagues, students, visitors. A very warm welcome to you. My name is Professor Pali Hanjin. I'm the head of the School of Medicine, Pharmacy and Health and the Dean of Medicine. And together with the Dean of Pharmacy, Dr. Andy Husband, it's our great pleasure to welcome you all here this evening to this very important and hallmarking inaugural lecture. Important and hallmarking because most of you will know that this is the first year of our new pharmacy program. It started only a few weeks ago after many years of planning. And this is indeed our first professorial inaugural lecture, something that I hope will be remembered, Stuart, for centuries to come. The arrival of pharmacy here is a very important development. We envisage that with almost 500 students at undergraduate level and with 100 or so postgraduate students, pharmacy will make a huge presence here at the Queen's campus. And the school itself, I think, will project itself from having been a nascent entity some 10 or 11 years ago to being one of the biggest departments at Durham University. But today belongs to Professor Stuart Wilson. Now, Stuart, who is starting his career again here in Durham, has been sitting nervously near the front, wondering how he was going to manage the rest of the evening. But I need to say a few things about Stuart. Stuart arrived here at Durham University from Dundee University, where he was attached to the big medical school and the Nine Wells hospital setup, which is known the world over for its research excellence and for its academic record. But he is really a native of Stockton-on-Tees. He's probably the only person I've known who's actually come back to Teesside. He actually went to Holy Trinity School not far from here. And he is the son of people who settled here during the heyday of industry, during ICI days when everything was golden and Teesside was one of the most advanced economies in the United Kingdom. Stuart has lived in many parts of the world. After going to primary school here, you moved to Holland, where you studied. And after that, you got your degree at Durham University and did a lot of postgraduate work in this locality. And I know that you've worked in Hong Kong, Glasgow, and many other places before arriving back here. And Stuart has many international connections. He has been an incredible catch for us, and what an appointment for us to have made. Someone who holds many fantastic grants and has done research at the highest level, and a distinguished MRC scientist and grant holder at the same time. But what are his real achievements? He's run the Glasgow Marathon. Not that I've ever seen anyone in Glasgow running, except to make sure the pubs don't close. He's climbed one of the highest peaks in the world in the Andes, and I didn't realize that until we got a few biographical details from him, and that's something worth talking about. Something over 6,000 meters. Now that sounds like it's heading for about 21,000 feet. That's quite substantial. Again, possibly an attempt to get away from Stockton. If you can't go sideways, go upwards. He's a concert-level pianist, and that is quite remarkable, and that's something we intend to capitalize on him. And believe it or not, he played Henry Miller in Calamity Jane. I think these are all accolades, and they show what a rounded person Stuart is. So it's my great pleasure to welcome you, Stuart, to give your inaugural lecture, a long-held tradition at Durham University for professors appointed here. And your title, as you can see, is Ion Channels in Health and Disease. But before you do that, I want to offer a particular welcome to Stuart's parents who are in the audience here. And I think that's tremendous. And I would also like to single out Dr. Bob Banks from the science department. Believe it or not, Bob was one of Stuart's lecturers when you started here in your BSc. And I gather that you're retiring within the next few months, if such a thing is possible for someone like you. And we I'm most grateful. It's an honor to have you here also. So over to you, Stuart.
Okay. Thank you very much for that very kind introduction. Okay, so the title of my talk, as Pali has said, is Ion Channels in Health and Disease. And to start this, I'm just going to give you a very little brief introduction, just the usual. Right, I'll have to do it this way. Oh. Okay, a very brief introduction, just to tell you what the ion channels are and why I'm interested in them, and then I'll come back and talk through the thing in more detail. But basically, to get the title out of the way, an ion channel is a specialized protein, and it's a protein which forms a pore in the cell membrane, which is the part of the cell which basically separates the inside from the outside. And these pores form channels through which ions can flow, primarily sodium, potassium and chloride. Now, this sounds sort of a pretty not very great news, but the important thing about this is all ions are charged. And here, the very important thing to remember is that anything with charge, if it moves through space, it will carry current. So ion channels are the basis of a branch of science called electrophysiology, which I think is one of the most fascinating and interesting branches of science. And it's fascinating because it's the area where physics and biology and, to some extent, chemistry meet. Now, so I might have a talk overview about what I'm going to talk about. I'm going to talk about the history of electrophysiology, how this subject developed and how it was discovered, and how it led to this idea that there were a separate class of membrane proteins called channels. Now, in giving a lecture like this, it's quite difficult to know what level to pitch it at. And because obviously there's a range of backgrounds here, from people with advanced knowledge of physics and chemistry, our students who have, I hope, very reasonable knowledge of physics and chemistry, and we have members of the general public. So I'm going to try and pitch most of the talk at a fairly low level, but at the same time, I can't really just completely ignore the specialists in the audience. So at 1 or 2 points, I am going to go into a little bit more detail about specialist areas, really to try and convey to people what is interesting and why this field interests me. I'm then going to talk on about a couple of areas, which I suppose you could call ethnopharmacology, the relationship between drugs and populations. And I'm going to talk about two very interesting things, which have both got to do with ion channels. The first is the problem of pufferfish poisoning, which is pretty much a uniquely Japanese problem. And the second thing is something that is perhaps a little less well known, licorice and high blood pressure. And as Pali said, I spent my teenage years in Holland, and this is a uniquely Dutch problem, which is quite interesting. It tells us something about ion channels. It also tells us something about pharmacology and about pharmacy. And then at the end of the talk, I'm going to give you a little taster of some of the work I've been doing in the last couple of years, looking at the role of ion channels in reproduction. So, to move on from there to the first part of our talk, the history of electrophysiology. Well, electrophysiology has a very, very long history. And we really, to start to look at this field, we have to go right back to Galvani, who is really credited with the discovery of electricity, full stop. Now, these days, we tend to think of electricity as part of the physical world. But Galvani actually discovered electricity as a biological phenomenon. And the way he did this was an experiment which was actually quite remarkable and also quite frightening. He basically took frogs' legs and he connected them, delivered electric shocks to them via lightning. So he used a lightning conductor and he delivered shocks to frogs' legs. And what he found was that if lightning struck the conductor on the building, that if we connected this lightning to the frog's legs, they would twitch and jump. And he concluded that there was a property of nature which he called electricity. And he later went on to show in more controlled experiments that we could get twitches in frog muscle if we brought the muscle into contact with different metals, zinc and copper being the classic two metals. And I'm sure the chemists in the audience would understand the significance of that straight away. But Galvani decided that there was some entity, something called electricity, that pervaded the universe, which provided us with a basic life force. And this idea was very popular at this time. Mary Shelley, of course, wrote the book Frankenstein. And the basic plot of Frankenstein is that Frankenstein brings a corpse back to life, or brings a corpse he has assembled back to life. And this idea seems quite fanciful to us now, but this was actually just the reflection of an idea that was discussed actively at the time. If electricity was the great life force, surely it might, ultimately, we might be able to find a way to bring the dead back to life. And this was an idea that was very seriously held. But I think the next development in this came again from another one of the great pioneers of physics, from Alexandre Volta. Now, Alexandre Volta, whose name, of course, we remember, the volt. Volta was best known as the discoverer of the battery. But once again, Volta was fundamentally, he saw electricity as a biological event, and he used his battery really to allow him to explore the effects of electricity on isolated frogs' legs in a much more controlled way. So by using his batteries, he could deliver very much more carefully controlled electric shocks to his preparations. And what Volta found was that the best way to make the muscles twitch was really to deliver electric shocks to the nerves. So Volta slightly stood back a little from this sort of idea that Galvani and the idea that was developed upon by Shelley and Frankenstein. And he said, no, electricity isn't a kind of a basic life force that drives things. Electricity is quite specifically concerned to the nerves. And he proposed that the nerves control the body by electrical phenomena. Now, this was quite a major advance at the time. Remember, at the time, people didn't really know what nerves were. We had dissected them out, we could know their layout, we knew their anatomy. But Volta actually made quite a major intellectual breakthrough where he suggested that the physiological function of the nerves was to control the body, and that the way in which this control was exerted was via some electric mechanism. And this idea was further developed later in the century by really one of the founders of modern physiological science, Émile Dubos-Réymond. And Émile Dubos-Réymond was very much a laboratory science, and he was very, very closely involved in the development of instruments that were sensitive enough to detect very, very small changes in the electric current. And what I've really tried to show here is I've tried to demonstrate his basic experiment in very simple terms. He actually dissected the sciatic nerve out of a frog, and he put a pair of electrodes in direct contact with the nerve surface, and then connected these to a very sensitive voltmeter. And the voltmeters that Dubos-Réymond were using represented the absolute ultimate of 18th century technology. And what he found was something very different to Volta's observation. What he did was to apply physiological stimuli to the nerves. And in the case of his classic experiment, what he actually did was to stretch the gastrocnemius muscle. And what he found was that when he stretched the gastrocnemius muscle, he could detect electrical activity in the nerves. So this had slightly changed the paradigm again. He was saying that there was a mechanism by which nerves generated electrical phenomena which could then propagate throughout the body. And so this is just really a slide that reiterates his hypothesis. He preserved that the function of a nerve fiber was to allow information to transfer between parts of the body. And by this time, the idea was well established that the function of the nerves was basically to allow the brain to control the body. So he'd really, by the end of the 19th century, we were in a position where this idea was very, very well founded, that nerves were in some way, there were electrical events which controlled the nerves. Now at this point, I'm just going to, until now I've been a little bit flippant just describing what a nerve is. I think here I'm going to introduce a little bit of anatomy here and just clarify what we understand by a nerve. When we refer to a nerve, we usually refer to one of the sort of physical structures that we can see if we dissect an animal or a human. And this bundle of tissue actually consists of very, very large number of cells and connective tissue. But in effect, what it is a bundle of single cells, single specialized cells, which are called neurons. So what we would call a nerve, and what Dubos-Réymond calls the nerve, was actually a bundle of many, many thousands of neurons. And each neuron was actually a single cell. And the important thing about a neuron is they're very, very unusual cells because they extend the entire length of the body. So there are some very, very large animals, giraffes, blue whales, this sort of thing. They literally have single cells which extend for meters. And in simple terms, we can look in very diagrammatic terms. A neuron really breaks down into three different parts. There's a part called the cell body. And the cell body is, if you like, the front end of the nerve. It receives inputs from sense organs, other neurons, and we now know that the function of the cell body is somehow to encode these inputs. Again, you get the right person, they could devote their entire lecture to this mechanism. I'm not going to talk about this any further. But the important thing is that once these inputs from different organs are encoded, the information is transferred the length of the axon. And irrespective of the size of the axon, that information is transferred to the other end. And then we come to the axon terminals, and these basically give us outputs. So they can send signals to make muscle contract, to activate other neurons. And one of the great intellectual challenges of the 20th century was to really try and understand the mechanism that allows information to pass from here to here. And really the problem with this stemmed from the fact that neurons, although they're very, very long, they are very, very fine. And in mammals, we typically find that neurons are about 0.2 to 20 microns in diameter. A micron is 1,000th of a millimeter. So, and these were really way, way too small to be in any way amenable to the technology available at the early 20th century. But however, as a generalization, we could say that invertebrate animals almost always have larger neurons than vertebrate animals. And there are many reasons for this. And in the early 20th century, somebody made quite a stunning discovery. And this was that squid, the standard animals that live in the sea that get fried up as calamari in many restaurants, actually have giant nerve fibers. Now, just to clarify here, we are not talking about the nerve fibers of the giant squid. We can get some huge squid. These are perfectly standard squid, up to a foot long. And they have a series of very, very large nerve fibers. And this discovery was made by J.Z. Young. And J.Z. Young was one of the great scientists of the early part of the 19th century, wrote many, many seminal textbooks. He certainly discovered this property of the nerve fibers of the giant squid. But J.Z. Young also has a more direct connection to Durham University because one of his PhD students was David Barker, who was professor of zoology at the University of Durham and a close colleague and associate of Bob Banks. So, and Professor Barker is well known to anybody who did biological sciences in Durham in the 60s and 70s. But J.Z. Young's observation, the discovery of that the squid had giant fibers, really unleashed a whole host of work where people used these because they were large enough to be amenable to the sorts of technology that were available at the time. And yes, this is actually just a little animation, shows what these fibers do. The physiological role of these axons is to coordinate the mantle contraction. Squid, as I'm sure you all know, they swim by jet propulsion, and they bring water into the siphon and then expel it into the mantle cavity, so this area here, and then it's expelled. And this expulsion of water and the jet propulsion depends upon coordinated changes to the mantle. And these fibers, which innervate the rear parts of the mantle, are huge. I mean, 0.5 to 0.8 millimeters in diameter. And they could be dissected out of squids and studied using the methods available in the 1930s and the 1940s. So how did people do this? Now I'm going to start a little bit more about the kind of nuts and bolts that goes behind some of these experiments. And how do we go about doing an experiment on a giant axon? Well, obviously, the axon has to be dissected out, and in a typical experiment, it would be attached to the bottom of an experimental chamber and be bathed with a salt solution that mimics the extracellular fluid. And the nerve will sit there perfectly happily isolated during this period. Now, to see what's going on inside the fibre, the other big development was the development of very, very fine glass electrodes, which could be used to penetrate the nerve itself. And by doing this, we get electrical access to the cell interior. So if we connect this electrode to an amplifier, we can record things going on inside the cell. The types of amplifiers that physiologists love to use are things called differential amplifiers. And the important thing about these amplifiers is they have two inputs, and they amplify the signal that is the difference between these two inputs. So if we use the fine glass microelectrode to connect our amplifier, to collect the positive input of our amplifier to the interior of the axon, if we connect the other input to the bath solution, and if we earth the bath, then we are effectively measuring the potential difference across the membrane of the axon. And what these experiments showed was that the inside of the axon was almost always negative with respect to the bath. And this is a very, very important property. They clearly showed and identified, which is now known as the resting membrane potential. So in the technical way we would describe this, we would say that the axon membrane is polarized. The axon has a membrane potential. The inside is slightly more negative than the outside. And clearly this was very exciting, because we'd known since the end of the 19th century that nervous activity had something to do with electricity. So this discovery that we had a standing potential inside the nerve was obviously of great interest to people trying to find out how these electrical signals are initiated and used. So just to display this, I'm putting this up just to stress this, that inside is more negative than the outside. Although this was first observed in nerve, it's a property of essentially all animal cells. And one of the big challenges at the early 19th century, late 19th, early 20th century, was to try and identify the physiological basis of this potential. Another property of these axons was that they were actually so large that we could physically squeeze their contents out. You could squeeze and collect the cell interior and analyze it. And these types of measurements gave us another very, very important property of nerves and of all cells. And I hope the students in the audience will recognize this slide, because I showed it in one of my lectures just a few weeks ago. One of the most, as I said at the time, one of the most important things that people can remember about cells, well, let's start with the extracellular fluid. Basically our blood and our extracellular fluids are basically sodium chloride. I'm not showing chloride here. So we have sodium, which is the component of salt, and very low levels of potassium. When we look inside the cell, and the squid giant axon was the first cell that was large enough to allow us to do this, we found something very different. Inside the cell was a very, very high concentration of potassium, but a very, very low concentration of sodium. So, and this is one of the most fundamental properties of all animal cells, that they sit basically in a solution of sodium chloride, but inside of the cell is basically potassium chloride. And this has very important consequences. It's a fact of chemistry that ions want to diffuse. If you have a concentration gradient, then any substance will try and run down that concentration gradient. So if we just think what would happen to sodium, we have a high concentration of sodium outside the cell, a low concentration of sodium inside the cell. So sodium will tend to diffuse down this concentration gradient. And a very, very important physicist called Nernst realized that this tendency to diffuse implied that there was a driving force acting on the sodium ion. The fact that the sodium ion was in a concentration gradient made it behave as if there was a force acting upon it. So sodium always wants to diffuse into the cell. But for potassium, remember the important point about potassium is that the concentration is reversed. So potassium always wants to diffuse out of the cell. So we have, if we just look at the chemical driving forces on sodium and potassium, they are opposite. But remember, the inside of the cell is also negative, and both sodium and potassium are charged. And another important point to remember is that any charged ion in an electric field will have a force upon it. Positive charges are always attracted towards negative charges. So there is a second force acting upon these two ions, and that is an electrical driving force. But in this case, they are both orientated in the same direction. Because sodium is positive, it always wants to move towards the negative area of the cell interior, and the same for potassium. Now, it was Nernst who first realized that these two forces could be combined, and Nernst actually went through the mathematics to express all of these forces in physical terms. And we can therefore pool these forces to give it something that we call the electrochemical driving force. And when we do this, we find that very different forces act on these two ions. There is a very, very large force acting on sodium, which always tries to move it into the cell. For potassium, the force acting upon it is a lot smaller. The net force acting upon it is a lot smaller, and it tries to move this ion in the opposite direction. So this is a situation that arises just from the chemical properties of these substances. But it was a very important scientist, Julius Bernstein, who realized that this may try and help us explain the resting membrane potential. And to try and illustrate Bernstein's thinking, I'm going to just represent the results of an experiment which was actually done later by J.Z. Young using the squid giant axon. And again, we have the same setup I showed before, the axon pinned out along the bottom of the bath, recorded to our recording equipment, and we're recording the voltage. So we're recording the resting membrane potential. And as we said before, the inside of the cell is always negative. But an important aspect of this experimental setup is that we can change the bath solution. So it's up to us. We can go to the lab, we can go to the chemical cupboard, and we can make up any bath solution we choose. So what happens if we exchange the normal bath solution? So we can, to start off with, the normal bath solution contains 5 millimolar of potassium, so we can plot a point on a graph showing the resting membrane potential measured at 5 millimolar of potassium. We can then superfuse the cell but lower potassium to say 3.8 millimolar. And what we find is that lowering potassium in this way always causes the inside of the cell to become more negative. And in technical terms, we would say that it causes hyperpolarization. So we can plot a new point on our graph relating to this potassium concentration. Then we can do the opposite. We can raise potassium. And here we find an opposite movement in the membrane potential. So we can plot another point. And then we can just complete the series. And what we can see is that the magnitude of the resting membrane potential depends upon the concentration of external potassium. So we have this nice, notice that this, the scientists and engineers amongst you might notice that this is a logarithmic scale. But we have this very clear relationship between external potassium and the magnitude of the resting membrane potential. And Bernstein rationalized these observations by making a very important and very influential suggestion. He suggested that the membrane is only permeable to potassium. So this implies that although there is this very large electrochemical driving force on sodium, sodium cannot move into the cell because the membrane is impermeable to this ion. Now, if the membrane is permeable to potassium, that means that potassium can leak out of the cell. Now, if we could just ask you to think about what's happening here, what we're doing is we're having a constant leak of positive charges out of the cell, which is not accompanied by a corresponding inward movement of positive charges. So the cell is always losing positive charge. And this effectively is the process. Bernstein suggested that this was effectively the process that established the resting membrane potential. Now, you may say that there's a big flaw in this argument. It won't last forever. The concentration gradient will eventually run down. And that would be a perfectly valid criticism. But the fact is, it's subsequently become apparent that these sodium-potassium gradients are actively maintained using metabolic energy. I'm not going into the mechanisms that underlie that. But what this means is that metabolic energy is basically being converted into electricity. By maintaining these concentration gradients and allowing potassium to leak out of the cell selectively, we are establishing an electrical system. So the cell membrane effectively behaves like a battery, or in more technical terms, it behaves like a potassium electrode. this is a picture of this conclusion was this hypothesis and much experimental data supporting it was published in 1912 by Julius Bernstein. And once again, I think it's instructive to just think about the barriers that Bernstein faced making these observations. I mean, he was using equipment that belongs to the realms of H.G. Wells. His experiments involved clockwork motors, electric shocks were delivered using induction coils, incredibly deep and insightful conclusions coming from really what are very basic, very difficult experiments. And it was really in the 1930s where J.Z. Young was able to directly measure membrane potential in squid giant axons that we had the first real solid confirmation of this. However, I won't go into Bernstein's other hypothesis. He did also predict a hypothesis to explain the conduction of the nervous impulse. And history has proved, although this was very, very influential and it drove thinking, his explanation of this was not correct. But so we're still left with a problem. Although we've seen a mechanism whereby the cell can establish and maintain a resting membrane potential, we haven't really addressed the question of how the cell can use electrical energy to send information up the axon. But this was, again, assessed using the squid giant axon. And to look at this, the equipment used got a little bit more complicated. We have the same recording system that I've described before, but now we impale the axon with a second electrode. And this electrode is connected to in simple terms, it's connected to a battery and a switch and a variable resistor. And the important thing about this is it allows us to inject current into the axon. So we can record voltage and we can inject current. Now, the relationship between current and voltage is, of course, enshrined in Ohm's law. So we can start to look at what happens. And here's the results of a first type of experiment. If I ask you first to look over here, if we just flip this switch and inject, so we're now displaying the results of a current monitoring system here along with voltage. And you can see that if we inject a very small pulse of current, we do see a deflection in the resting membrane potential, as you would predict. Important point, though, is if we look at the resting membrane potential measured at a site that is remote to the site of current injection, we see no change. So this tells us something very important again. It tells us that that nerve is a very poor cable. If this was a cable, the sort of cable that we use to send electricity around the house, any change in voltage here will be reflected here. But it tells us the nerve is a very poor cable, which really just dispenses with the idea that cable transfer of electricity is important. If we repeat the experiment and inject a slightly larger current pulse, as we would predict from Ohm's law, injecting more current causes a larger deflection of voltage, but we still see no change here. So although we've disturbed the resting membrane potential, we haven't done anything that would allow us to transfer information the length of the nerve. However, if we change the injected current even further, something very, very different happens. We see that we inject current, we see that the resting membrane potential shifts again, but then suddenly we see this very rapid transient. And the important thing about this transient is that we can record it at a site remote to the injection. And when these experiments were done more carefully and consistently, what people did was they identified something which was called a threshold potential. And it seemed to be a property of neurons that whenever the resting membrane potential rose above the threshold potential, something happened which led to the generation of this signal which could travel the length of the axon. And this is just the classic drawing of an action potential. We see the resting membrane potential here, plotted against time. An action potential consists of a very, very rapid change in potential. It goes from a negative value right up to a positive value. And look at the time scale here. This is about 10 milliseconds. So this is a very, very rapid event. It's followed by a repolarization, and almost invariably the resting membrane potential, it undershoots the resting membrane potential, and then we see a slow return to the normal condition. Now, the important thing about these action potentials is that they were all or nothing events. If I'm just, yeah, sorry, they were all or nothing events. if we looked at a range of different neurons, then each different neuron may generate action potentials that are slightly different from each other. They were individual, but within a neuron, a particular neuron would generate action potentials that were identical to each other. And the second important, so basically whenever we change the membrane potential in such a way to cross the threshold potential, we see an action potential. And as I say, the most important thing about this is that it propagates along the length of the axon. So if we imagine that we initiated an action potential by injecting current here and then measured voltage at a series of sites along the length of the axon and plotted the output from these measuring devices against time, we would see the same event at different times. So this was kind of like a ripple of electricity that ran the length of the axon. And what we show here is the first direct recording of an action potential. And there were two groups who succeeded in doing this, Curtis and Cole, who were based in the Woods Hole lab in Massachusetts, and Hodgkin and Huxley, who were based in Cambridge, and who did most of this work at the Marine Biological Laboratory in Plymouth. Obviously, in those days, if you wanted to get squid, you had to go to the seaside. And the Marine Biological Lab in Plymouth. There are a number of features about this recording I'd just like to point out to you. The first is that it is clearly recorded on an oscilloscope. That's obvious to us. In actual fact, one of the problems of making these recordings, you can see that we're recording, well, this scale is in millivolts. So we're recording a signal that's about 80, 90 millivolts, possibly 100 millivolts, and a very brief signal. And one of the big problems about making these recordings was finding recording systems that were sensitive enough to do it. And it was suggested that why don't we try and hook the output up to what was then known as a cathode ray tube. And in actual fact, this was the first use of the oscilloscope. The oscilloscope was actually invented to record these signals. There were a number of problems. The problem initially was to generate consistent enough voltages to drive the beam with steady speed across the time axis, which led to problems in calibration. These problems were solved by connecting a second channel, which was connected to a very, very sensitive microphone. And what we see here is the microphone listening to a tuning fork. So to calibrate the time base, we recorded the output from a tuning fork with very, very high fidelity. And notice as well the timing of these recordings, 1939. A year later, there was a similar need to record very, very rapid and very small electrical events. These were radar echoes of aircraft. And most of the people who developed this equipment at the outbreak of war, R.V. Jones identified them, and they made a huge contribution to the development of radar, because they were the only people in the country who were consistently recording voltage deflections of the sort of same magnitude that the radar engineers wanted to characterize. So again, huge development technologically. And as I say, just to reiterate, the axon potential is a predictable deviation from the resting membrane potential. In a particular nerve, the action potentials are identical, and it is quite remarkable to see them. They're not just identical, they are so identical, it's unbelievable. It's an all or nothing event, and it is the action potential that transfers information along the nerve. And just to reiterate again, these action potentials are seen whenever the resting membrane potential crosses the threshold potential. So this discovery really focused attention. If we wanted to understand how action potentials worked, if we wanted to understand how nerves conducted information, we had to understand the way in which what happened when we crossed this threshold potential. And the bottom line, although developments were made and there were a number of important observations made, there was really no way of addressing this in the pre-war years. And the way around, and all of the pre-war experiments, as I've tried to illustrate, they involved injecting current and watching what happened to the voltage. In the post-war period, there was a huge technical and academic breakthrough that basically reversed this process. What we effectively do is inject voltage and measure current. And this is called voltage clamp technology. Now, this is part of this talk where I do apologize. I'm going to try and explain how voltage clamp circuit works. this really is something that really is aimed at physicists and engineers, but I think it's important. It's an important part of the talk, and I would like if you don't understand, if you could just bear with me a little moment. Again, we have our axon set up pretty much the same way we did before. We have an electrode for injecting current. We have an electrode set up for measuring voltage. It's connected to an amplifier, and the bath is earthed. The most important thing we have to add to this setup now is a battery, a variable resistor. So we have a battery, a variable resistor here. And what this does, by adjusting this variable resistor, we can generate any voltage we want. We can generate a reference voltage, we would call that. So if we decide we want to generate 30 millivolts, we can adjust this equipment to get a 30 millivolt signal here. Now, the real stroke of genius here was in this step, where they took the input-output from this amplifier, which, remember, is giving us the value of the resting membrane potential, and fed that into the inverting input of a second amplifier, along with the reference voltage. So this amplifier is giving us the difference between the resting membrane potential and our reference voltage. So if we now connect this amplifier to a current voltage converter and appropriately scaled, what we can do is we can inject sufficient current, we inject a controlled amount of current back into the membrane. And this is essentially a feedback system. What does this do? Really, what we can do, if we imagine imposing a step change of voltage here at our reference electrode, if this all works, and if it works fast enough, then we can impose a step change upon the resting membrane potential, and we can record the current required to make that change. Now, I'll go on to say why this is importance of this physiologically, but once again, I think it's worth stressing that this equipment was built in the immediate pre-war years, and we're all used to the idea of amplifiers now. Remember that in these days, transistors simply did not exist. And what's more, it was also in post-war austerity Britain. And this equipment was built really with valves scrounged out of radios. It was designed, built in-house, an amazing achievement. So yeah, so really what voltage clamp technology lets us do is control the membrane potential and record the current. And this is just really to show another level of complexity was introduced into these experiments, that they also superfuse the inside of the axon. So this is the experimental setup that Hodgkin and Huxley and their collaborators were working with in Plymouth and Cambridge by the late 1940s. Well, so what? What does it let us do? Well, if I can just put this little model here, which, just to remind you, this is kind of Bernstein's view of the membrane. And the important properties of this is that the membrane is impermeable to sodium, so although there is a very large driving force on sodium, it can't get into the cell. There is a smaller driving force on potassium that allows a constant leak of potassium out of the cell. And what we show here is a step change in voltage from, in this case, from minus 103 millivolts to these test voltages imposed using the voltage clamp circuit. I should just actually like to say at this point, I'd forgotten, all of these records I'm actually going to show you are experiments done by Stephen Mansell and I. We were there 50 years later than Hodgkin and Huxley, but when we were setting up the experiments, really as a student teaching exercise, we repeated a lot of their classical experiments. And the important thing here is we're showing membrane current. See, now it's plotted as microamps. And we can see basically that this step change in voltage causes no change in current. Again, just a little aside to physicists and engineers amongst you, you may have noticed, you may actually be in the process of thinking, well, that's got to be nonsense because you've changed the voltage. Ohm's law tells you must change the current. There's another technical refinement that they introduced, which was basically these passive currents evoked by just ohmic current flow was subtracting away. So really, when we see a flat line like this, what we're actually saying is that there was no deviation from Ohm's law. We're not saying there was no absolute current. There was no deviation from Ohm's law. So basically, what that is telling us is the physical properties of the membrane had not changed. So depolarizing voltage steps to potentials that were below the threshold threshold potential had no effect on the properties of the membrane. However, when we depolarize the membrane to a more positive value that is above threshold, something very interesting happens. And this is just one of many results that they presented. This is the most important. We see a sudden transient development of current which decays away. And what we show when Stephen and I did this experiment, we, as Hodgkin and Huxley did, we recorded under control conditions, and then we switched the bath solution to one containing virtually no sodium and repeated the experiment. And you can see that this disappears. So Hodgkin and Huxley rationalized that what happened is that once we depolarize past threshold, the cell membrane suddenly becomes permeable to sodium. And this is really reiterated in the next slide, where what I'm showing here are the physical properties of the membrane under resting conditions and the physical properties of the membrane that have been depolarized above threshold. Below the threshold potential, Bernstein's theory applies. The membrane is permeable only to potassium, and we have a resting membrane potential. Above the threshold, we suddenly have an increase in permeability to sodium. So now, whereas before we had a very small leak of positive charges running out of the cell, now we suddenly have an inrush of a very, very large number of positive charges. And I have just noticed, looking at this slide for the first time, that I've forgotten to change the membrane potential. That should say plus 20. So basically, this was Hodgkin and Huxley's main conclusion, that the event that happened as we crossed threshold was a sudden increase in sodium permeability. And they rationalized this by predicting the existence of a family of entities. They didn't know what they were, but they called them voltage-gated sodium channels or voltage-gated ion channels. And the important point about this hypothetical entity was that it was a pore in the membrane which was permeable only to sodium, and by inference there must be other pores that were permeable to other ions, but that this pore had in some way a gate on it, and that if the membrane potential was below threshold, this gate was closed, so sodium couldn't penetrate the cell, so Bernstein's model could operate normally. We could have this leak of potassium establishing the membrane potential. Once we crossed threshold, this channel would open and allow sodium entry. And this, in essence, is Hodgkin and, And the important point about Hodgkin and Huxley's theory, when they thought about it, the entry of sodium is in effect, it's exactly like the experiments where we're injecting current. Sodium entering the cell is carrying current into the cell, so it will slightly shift the voltage. And if we open a channel here, we'll slightly shift the voltage, and this channel here, will suddenly the membrane potential will rise above threshold here, so we'll get sodium influx, which will then affect the potential next to it. And we'll get this ripple that runs down the membrane of transient openings of sodium channels. And this, in essence, is Hodgkin and Huxley's theory of nervous transmission, which was published in the Journal of Physiology in 1952. And it was this work that they won the Nobel Prize for Medicine and Physiology in 1963. So this was, without doubt, one of the great intellectual achievements of the 20th century. It was subsequently shown to be applicable to essentially all all mammalian nerves. So everything that happens in your nervous system is dependent upon this process. And in a little bit, I'll show you a little bit about just how important this is. Now, these sodium channels were predicted by Hodgkin and Huxley as hypothetical entities. But amazingly enough, technology has moved on to such an extent that we are now able to directly monitor current flow through single channels. And this is shown here. Notice the scale here. This is membrane current in picoamps. A picoamp is 10 to the minus 12 amps. And we can see what we see here. It's basically is membrane current, and we can see these abrupt transitions. And what this is, and it's really described in this paper, is that these are current flow through individual channels. And I find this quite a remarkable observation, because really what we're showing here is a single molecule. It's a large molecule, admittedly, it's a protein. But it is the large protein that is in thermal motion and is basically flipping between two metastable states, one of which is able to conduct ions and one of which is not. And I'm sure, well, Hodgkin and Huxley were alive and witnessed this development. Neher and Sackman, who drove this, won the Nobel Prize for Physiology and Medicine in 1991. So a huge development. And without going into it too much, one of the questions about this is how could it be that we have these unitary channels whose behavior is not really determined? How could they give rise to predictable changes in conductance? And the answer is shown here is basically there's an awful lot of them. So what we show here are a series of sweeps where we've done exactly the same experiment that I described earlier. We just imposed a depolarizing voltage pulse on the cell. And you can see if we look here that we're looking at the behavior of individual channels. And you can see that basically, although channels do open, there's no real predictable way. But when we pool these results together and work out an averaged response, we can see that we start to see a predictable response, which looks like that we've quantified in a neuron. So, and again, there's a huge amount of science gone into this. How do these large numbers of very small channels that display stochastic random behavior give rise to predictable behavior in cells? Well, since then, many different types of channels have been identified, they've been cloned, expressed in cells. Sodium channels, potassium channels, chloride channels. They're found in all cells of the body and they play many roles in physiology. And most importantly, many diseases, many genetically transferred diseases arise directly as a consequence of genetic changes that affect proteins which encode channels. And you can talk about channelopathies, a whole class of diseases. And I've just named some of them here. Myotonia congenita, Bartter syndrome, cystic fibrosis is possibly one of the most... I'm sorry, cholera really shouldn't be there, actually. Cholera is caused by dysregulation of a chloride channel. So that really concludes the first part of my talk. And really, I'm going to go on now to talk a little bit about this sort of ethnopharmacology and just a couple of compounds which I think are quite interesting, compounds that interact with channels and give us some indication of how these channels are important. And the first thing I'd like to talk about is this pufferfish poisoning. Now this is a picture of a pufferfish swimming around perfectly. And the thing about pufferfish is that they contain a toxin, tetrodotoxin, TTX, and this toxin is shown here. And this toxin is extremely lethal. Ingesting about 25 milligrams of TTX will almost certainly be lethal. It causes paralysis and respiratory arrest. So you become paralyzed, you can't breathe, you suffocate and you die. But despite this, the pufferfish is a delicacy in Japan where it is known as fugu. And here are fugu for sale in the Japanese fishmongers. Now in Japan, in any 1 year, the authorities will have to deal with about 30 fugu poisoning incidents. And this sounds not too bad until you realise that fugu is a delicacy and is usually only served at weddings and things. So a fugu poisoning incident can involve up to 60 people. Now, the death rate from fugu poisoning is about 7%, which sounds quite good. This is 7% in Japan. In Japan, doctors are aware of fugu poisoning. They are trained to deal with it. They recognise it. The death rate from fugu poisoning in places like Toronto, well, Toronto is starting to recognise it. In other parts of the world where you have a Japanese expat community is much higher. Basically, tetrodotoxin is metabolized. Although the person is completely paralyzed, if you can get them into an intensive care unit and onto a ventilator and keep them alive for 3 or 4 days, hopefully the body will degrade tetrodotoxin and the person will recover. And that is the only treatment. There is no antidote. There is no cure. And now, most of the TTX is actually found in the fish's liver. Some of it's found in other parts of the body. Some of it is found throughout the fish. And in Japan, restaurants that serve fugu have to be specially licensed and they're inspected, very strict inspection regime. All the knives and implements are kept separately. Fugu chefs have to be separately trained and licensed. And most Japanese restaurants are very safe places to eat fugu. They're very tightly regulated. It really, the problem arises, and it's not even because of enthusiastic amateurs, fishermen who catch fugu and try and clean it themselves. The problem arises because a lot of people choose to eat the liver. for reasons that are known. And apparently, if you eat just the tiniest sliver of fugu liver, your mouth and lips go numb. And this is regarded as, as something that's good. And perhaps the best example of this is provided by this gentleman, Bando Mitsugoro. Bando Mitsugoro was one of Japan's best-known actors in the classical Japanese tradition. He was described as a living legend. And the way in which Bando met his fate, he was in a fugu restaurant, and he and his 3 companions said to the chef that they wanted to eat a slice of pufferfish liver. And the chef initially refused, but he was put under pressure, and because Bando was such a national figure, he eventually complied and agreed to give them 3 very, very small, very thin slivers of pufferfish liver. His 3 companions refused to eat theirs, and Bando, we presume fueled by sort of sake-driven machismo, said that he would eat the lot. And he died within 17 minutes. And this did inspire change in Japan. The sale of pufferfish liver was expressly forbidden in direct consequence to this incident. But what does tetrodotoxin do? Well, the structure of tetrodotoxin really became apparent at about the same time that Hodgkin and Huxley's model of the nerve became known. So obviously the first thing people did was find, what does it do? And here again is one of Stephen Mansell's experiments, which really reiterates exactly what Hodgkin and Huxley did. We show again a voltage clamp with step depolarizations to above threshold, and you can see that under control conditions we get these nice little inward currents, exactly as you would predict. What we show here is after exposing the cell to I can't honestly remember the concentration of TTX, but it was vanishingly low. When you buy TTX from Sigma, you look at the vial and you think there's nothing in there, and you kind of dilute it out and put it onto a nerve, and it blocks the sodium current. And what TTX does, it's beautifully designed by nature to actually bind to these voltage-gated sodium channels. So TTX effectively plugs the channels that prevent sodium entry. Interestingly, the pufferfish itself has a mutant form of the sodium channel. TTX actually originates because of bacteria which the fish ingests, and these are normally excreted, but the pufferfish has chosen to accumulate them as a poison. And it can do this because it has an evolutionary, well, in its case, very strongly conserved mutation. But TTX effectively binds these voltage-gated sodium channels and gives us complete disruption of nerve function. Now, another feature of TTX is that it can't cross the blood-brain barrier. So whilst it may make you lose consciousness, it may make you collapse and stop breathing, it does not make you lose consciousness. So it is a very, very dangerous toxin. It's an interesting example of ethnopharmacology, but also the discovery of TTX and the identification of its mode of action went a long way towards verifying Hodgkin and Huxley's model of nerve transmission. So a bit more ethnopharmacology. Licorice and high blood pressure, a uniquely Dutch problem. Well, we're now moving into a completely different organ, and in very simple terms, we're talking about the kidney. And one of the most important functions of the kidney is to regulate the amount of sodium in the body. And it's become apparent that this is also dependent upon a sodium channel. It's a different type of channel to that found in nerve. It's called the epithelial sodium channel, which is almost abbreviated to ENaC. And basically, the kidney is set up in slightly a strange way. Lots of sodium enters the urine, and it's then recovered and recycled back into the blood. And any sodium that isn't recovered here is lost with the urine. So this is one of the fundamental mechanisms that allows the body to regulate the amount of sodium present. And we know this is important because a couple of genetically transferred diseases. There's a very rare disease called Liddle's syndrome, and this is an inherited form of very severe high blood pressure. And we now know that Liddle's syndrome is caused by mutations in the ENaC that make this channel overactive. So irrespective of the amount of sodium in your diet, you are constantly recovering sodium from the urine. And so we get fluid retention and very high blood pressure. And there is also an opposite disease, which is caused by inactivation of ENaC. And this is called pseudo-hypoaldosteronism. ENaC is underactive, so we get virtually no sodium retention. So people with PHA are continually losing sodium and water. So these are both caused by genetic changes in this channel. Another important point about ENaC is that it's controlled by hormones. Suppose we have low levels of sodium in the diet, your body would respond by secreting a hormone called aldosterone. And aldosterone has the effect of upregulating this pathway and allowing more sodium to be recovered. So this is one of the very, very important mechanisms that allows us to regulate the amount of sodium in the body. Now, anybody who spent any amount of time in Holland will know that the Dutch love licorice. I don't know why. They love very, very strong licorice, which is actually frankly disgusting. They eat loads of it. And licorice actually contains this compound, glycyretic acid. And glycyretic acid, unfortunately, it's not a very good drug, but it is at very high doses. It is able to mimic the effect of aldosterone. And so we can get consumption of large amounts of licorice can lead to fluid retention and high blood pressure. And practically the only population that we see this problem in are the Dutch. So another example of ethnopharmacology. Well, in the last few minutes, I'd really like to just talk a little bit more about what I've been doing in the last few months, the last couple of years. And our work here has really been influenced by this gentleman, Chris Barrett, who I met just a few years ago. Chris Barrett is the Professor of Reproductive Biology at the University of Dundee, and he's got an interesting career. He initially trained as an embryologist, worked in an assisted conception unit, became very senior and became fascinated by assisted reproductive technology. He's been a member of the Human Fertilization and Embryology Authority, so he's one of the key figures who determines who can and do what. And by his own admission, he attempts to convert every scientist he meets to reproductive biology. And this is really how I ended up in this field. This is exactly how I ended up in this field. Well, we're going to just a little bit about sperm. We all know that sperm are motile cells that fertilize the ova, but actually sperm have very strange patterns of mobility. What we see here is a human sperm just swimming along quite happily. And this we call progressive motility. Now, the problem with sperm that display this sort of progressive motility is that they are absolutely unable to fertilize eggs. And to do that, sperm have to undergo a change to another pattern of mobility which we see here, a hyperactive motility. And you can see that the head wiggles around. And what we show here is just obviously some paths tracing out these patterns of motility. And this development of this hyperactive mobility normally occurs within the female reproductive tract, and it occurs in response to progesterone and other female factors. And this is actually why IVF was so difficult to achieve. If you think about it in simple terms, you've got sperm, you've got eggs, you put them together, they fertilize. You know, what's so difficult about in vitro fertilization? When it actually came to the practicalities, it simply didn't work. If you just put sperm and eggs together, you will not achieve fertilization. And Bob Edwards' real achievement was that he systematically went through the biology of sperm and systematically worked out methods that you could keep sperm in vitro in the lab and bring them into this sort of condition where they could fertilize the egg. And IVF still has a very low success rate, and many of these problems stem from male participants. We tend to think of IVF as being something that was developed to overcome problems of fallopian tube defects and this type of thing. But when you actually start doing IVF, you find that actually, it's the problem is the other way around. It's the male participant. And Chris Barrett has spent an awful lot of time looking at these patterns and this is some of his recently published work. And what we see are these patterns, sperm characterly, they undergo all sorts of, they swim along in progressive motility for a while, and then go into this hyperactive motility and then swim off. And really to a very brief summary of this paper, which Chris has recently published, they very closely studied these patterns of sperm motility under conditions that would mimic the situation we see in the female reproductive tract, and starting to see correlation of these patterns of motility with the ability to achieve conception. And the other thing that's becoming increasingly apparent, mainly from work of a group in California, is that hyperactivation does involve changes in ion channel activity. And it was really for this reason that Chris attempted to persuade me to be a reproductive biologist. And this is something I never would have thought possible. What we see here is a single human spermatozoa, and this out of focus thing coming up here is actually an electrode attached to the sperm. So we've gone from the state where Hodgkin and Huxley were having to find giant fibres to do these types of experiments to now recording currents from single sperm. And what we show here are responses to depolarizing voltage steps, where we can see the development of this, is outward current. And again, note the scale here. We're seeing about 120 picoamps of outward current, so 120 times 10 to the minus 12 amps of current. And we've shown that these currents are carried by potassium. And we really just work which has just been published in accepted in human reproduction. We've identified components of this response which are influenced by progesterone, and so we feel that we are making some progress into understanding the sort of biophysical events that accompany hyperactivation. We've also, interestingly enough, explored the extent of environmental pollutants. I'm sure you're all aware of stories in the newspaper about declining sperm quality. And one of the things we found was that this compound, DDE, which is a metabolite of DDT, which persists in the environment and has been blamed for loss of sperm function, does have direct effects on spermatozoa themselves. But I think possibly the most exciting, and this is very, very speculative almost, is that what we show here, again, membrane currents, they're recorded using a slightly different protocol. Instead of bringing voltage steps, we just put a voltage ramp. But what we show here are typical currents that we record from a donor. So if we get a normal volunteer donor to come into the lab, and we treat their spermatozoa in exactly the same way they would be treated in an IVF clinic, we can record a current like this. These are 4 sperm from patients who were unable to conceive naturally, but were able to fertilize oocytes in vitro, although only at very low rates. And it's always like anything at the beginning of our project. They all have slightly smaller currents than this one. But this gentleman here is what really interests us. This chap had a clear motility defect. He did produce sperm. The sperm were motile, but they were unable to undergo this hyperactivation response. They've had defects which Chris had identified, but most excitingly, we see this total loss of potassium current. I mean, this I really cannot stress enough. This is ongoing work. I'm not making any conclusions about that. But the important thing is that we're starting to link these physiological studies of sperm and link them up with clinical observations. Okay, so that's really my talk. But as Pally said, it's not the first time I've been at Durham University. And I thought it might be interesting to show some of the students here. This is the science prospectus for the University of Durham, 1975 to 1976. And what we show here are labs in progress. Here students are measuring the respiratory coefficient using the Warburg apparatus. And it's quite clear that you do not have Dr. Harburn telling you to put your lab coats on. And this is an experiment I can remember doing as an undergraduate, photo from the prospectus. And what this student is actually doing is measuring an action potential in the cockroach ventral nerve cord. And I can remember doing that experiment at the time. So that really, this very experiment, probably with that very equipment, was one of the first electrophysiological studies I did. So many thanks from the 70s. It would be unfair to single out many people, but David Barker, our head of department, Ken Bowler and Chris Jones, who I did my PhD with. The 80s, David Yadulovich, Geo Mann, Hugh Elder, David Jenkinson, particularly Grant Butt. Grant Butt and I did first big electrophysiological studies together when I visited him in New Zealand. The 90s, people I've worked with, Patrick Wong in Hong Kong, Richard Olver, Dafford Walters, Chris Barrett, and of course all my students and postdocs who have worked with over the years. So thank you very much.